Hello, everyone. My name is Marleah, and I am an undergraduate researcher with Dr. Rita in the biology department here at West Virginia University. Today, I will be sharing my project, a comparison of peritrophic matrix-associated gene expression between Tsetse species of varying vector competence. African trypanosomiasis, more commonly known as African sleeping sickness, is a disease that is endemic in 36 African countries, according to the World Health Organization. African sleeping sickness can result in meningoencephalitis, and, if left untreated, death is likely. African trypanosomiasis is caused by parasites known as trypanosomes, which are transmitted to human hosts through an insect known as the tsetse fly. However, tsetse fly species have varying vector competence, or abilities to sustain and transmit the parasite. Therefore, it is important that we understand how vector competence is determined. In order to answer this question, we compared the expression of genes in the midgut of tsetse flies, focusing on the components of the peritrophic matrix, or PM, which is a structure made of chitin, and is pictured in the fly diagram in Figure 1. The PM acts as a protective barrier between the gut epithelium, or lining, and the blood meal, which is the sustenance of the tsetse fly. There are two types of PM found in insects, and the tsetse fly has a type 2 PM, thought to be produced in an organ called the cardia. However, recent work has suggested that PM synthesis can occur in other biological structures within the tsetse fly. We examined two species of tsetse fly, Glossina brevipalpus, which shows low vector competence, and Glossina morsitans, which displays high vector competence. We hypothesized that G. morsitans, as it is more likely to sustain and transmit trypanosome infection, may show the downregulation of genes involved in the synthesis of the PM. The idea behind this is that the PM plays a role in the tsetse fly's immune defense. When a tsetse fly consumes the blood of an animal infected with a parasite such as a trypanosome, the PM acts as a physical barrier between that infection, which exists in the blood meal, and the rest of the organism behind the gut epithelium. Therefore, it makes sense that a compromised PM will facilitate more trypanosome infections. In order to test this hypothesis, we followed the steps outlined in Figure 2. We dissected and homogenized G. morsitans and G. brevipalpus adults that had just emerged from their pupal states. We isolated the nucleic acid RNA and constructed a complementary DNA library. We then sequenced these libraries with Illumina technology. 123 orthologous genes, or genes shared between two species due to common ancestry, were identified and classified into various cogs, or clusters of orthologous groups, which detail the function of a gene. The breakdown of genes by cog is shown in Figure 4. 47 of these orthologs were differentially expressed between tsetse flies, shown in Figure 3, and of these 47 orthologs, 14 were found to have high expression in either G. morsitans or G. brevipalpus when compared to the threshold expression of an alpha-tubulin gene of reference. The expression of these 14 genes are displayed as a heat map in Figure 5, with genes of higher expression displayed in yellow. These results support the idea that the PM is not solely synthesized by the cardia, but that midgut gene expression may contribute to PM synthesis as well. Of the 14 highly expressed PM genes, 71% occurred within G. brevipalpus, supporting a more robust PM, and therefore likely contributing to the low vector competence of the G. brevipalpus species. However, 47 orthologs were differentially expressed, which absolutely merits further investigation, and future work might include microscopy analyses comparing Teneral G. morsitans and G. brevipalpus flies to determine whether expression differences translate into phenotype by taking physiological measurements such as PM thickness or physical composition. If we can understand the differences between tsetse fly species, G. morsitans and G. brevipalpus, this will also give us an understanding of the differences between high vector competence and low vector competence, and in what genes might promote or prevent the sustaining of trypanosomes within the fly. These results may be helpful in determining the new designs of interventions towards vector control of tsetse flies and their transmission of African trypanosomiasis. Thank you for your time, and any references can be found in the description of this video or emailed upon request. If you have any questions, please feel free to leave them in the comment section of this video, and have a great day.